It's a great pleasure for me to introduce Professor Derek Lundberg, who's today's GetGenome Seminar speaker. Derek studied plant microbe interactions in Arabidopsis thaliana roots during his PhD with Jeff Dangle at UNC Chapel Hill in the US, and primarily used amplicon sequencing to classify and quantify bacteria. In 2014, he moved to Germany as a postdoc in the lab of Dietliff Weigel at the Max Planck Institute of Biology, studying primarily leaf-associated bacteria in field-grown and wild plants. He further developed expertise and interest in diverse sequencing methods, including more genomics and metagenomics. Excitingly, in 2022, he moved to the Swedish University of Agricultural Sciences to start his own group, focusing on the fundamental science of beneficial bacteria interactions with plants. So I think this is an area that many of our members are very interested in. So Derek, the floor is yours. It's a great pleasure to welcome you.
Great. Thanks, James. I'm really glad to be here. I learned about GetGenome when Sophie and Kamun from Norwich visited SLU, where I work. And I was especially fascinated to get involved in this effort because I guess it's largely because my wife is from Bangladesh and I've been to Bangladesh, and I've seen how agriculture there can work. Like, it's amazing how fertile the soil is. Like, you cut a branch off a tree and it falls on the ground, and now you've got new plants, or the fence posts start making trees suddenly. So it's just a different world than some of the colder places that I've lived, and I've met a lot of the people there too. So, and I just feel like there's just a lot of really smart people, and the resources that GetGenome provides, I think, is just an excellent project. So what I'll talk about today is kind of what we're trying to do in my group at SLU, and I'll first explain a bit how I got there. And James already walked through the biography, so I can skip a lot of that. But I grew up in the United States, and since 2014 to 2022, I was working in Germany, and now I moved to Sweden. And I was first looking at root microbes, and then leaf microbes, and now finally I'm looking at leaf and root microbes, and I'm really focusing a lot on one bacterial genus for several of the projects in our group, and I'll explain why next. But as we move forward, there's a real need, a well understood need, for sustainable agriculture, as opposed to agriculture where we're using methods that are damaging to the environment. So in this picture, this is just a photo I thought was really striking. It's a dry field. It's a quinoa field. You see, climate change in action, and you also see somebody spraying pesticides without protection. And this is all kind of the thing that we want to avoid. We want to grow better, more crops, but not in a way that puts people or the environment in danger. And actually, this is where my research interest and field comes in, because microbes, and in my case, I'm studying bacteria, can contribute to sustainable agriculture. And the way this works is basically you've got microbes that live on plants, in the roots, on the leaves. Some of them are bad. These are the little devil emoji bacteria here. And there's some that we don't know what they do, so I've got just a straight face. And then there's also some beneficial microbes. And one way they can be beneficial is they can fight the pathogens. They also have ways to bring nutrients to the plant. A famous example would be rhizobia, that can fix nitrogen in the roots of some plants. There's ways that the bacteria can degrade toxins in the soil, for example, disinfectants or other types of recalcitrant chemicals in the soil. Bacteria sometimes have pathways to make that go away. And also there's different plant hormones that they can make. And the net result is that the beneficial microbes help the plant grow better. And of course, an advantage when you're spraying anything on a plant, whether it be a chemical or a bacteria, is that you can apply it basically to any plant, whereas breeding is a slower process, of course, very powerful, but slow. And microbes are also less regulated usually than chemicals because they're already in the environment. And another potential advantage is a microbe, if it can live on the field plant, it can persist there, whereas a chemical might need to be resprayed. If you happen to have a beneficial microbe that can survive on the plants, it can continue to be beneficial. And the net result would be that there's, in the end, fewer chemical fertilizers and pesticides. Not necessarily a practical replacement for all of these chemicals, but microbes are part of a sustainable solution. And that's well recognized these days in that many major companies in the agricultural industry are funding huge programs to try to market and find these microbes. And some of them are successful, but the problem is this is an inefficient process. Like this is some data from an industry paper, actually written by members of one of these companies, and a typical situation would be they start with many thousands of isolates. Maybe they test them in the agar plates, test them in the growth chamber. They find some that seem to work well, but many don't. So, and then they can move some of them onto the greenhouse screening. But then, some fail there. Then they can do some field trials. In the end, so many things stop working about these beneficial microbes that you have one or two product candidates, and then you've got to try to approve them. And it's just a slow needle in a haystack kind of to find them. And that's what I want my research program to help solve. And the idea is, instead of like looking for strains that are successful in the lab and following this pipeline, which is super labor intensive and really beyond what a small resource academic lab like mine could do anyway, is rather to study bacteria that are successful, figure out how they're successful, and then this knowledge can be used ideally to then help find better beneficial microbes and to make this process more efficient. And to that end, I think I found a microbe that's going to give a lot of the answers. And this microbe I came across with some experiments we were doing in Germany. And those experiments were basically, we went to a site where Arabidopsis thaliana was growing. This is a kind of a cold weather European plant most of you are familiar with. And it's these ones with these white flowers here. And it's just really, of course, well-studied plants. And we were looking at this plant in the wild, but also looking at all the other plants around it, because these plants are annuals. In the summer, they die. And I was just curious what happens when the Arabidopsis dies, when there's just seeds left. Where do all the microbes go? So my approach to that was basically to go to this field site and to collect not only Arabidopsis, but also many of the other plants that were there and find out what microbes are on those plants. And we just collected anything that we could recognize was really common at this field site. Sometimes we didn't even know the botanical name, so we gave it some kind of just a closest abbreviation here. So that's what these stars mean. And we collected in the spring when there was still Arabidopsis present, and then also in the late summer when the Arabidopsis was totally gone. And then one of the first things then that we did was just look at the community of microbes through a standard method, the ribosomal sequencing. And in this chart, what you see in this bar up here, these colors represent the different plants that we collected. And you can see Arabidopsis in the light green and some of the Brassicaceae in the other colors of green. And in the bottom part, you see bacterial families. And basically the proportion of each family that can be found on each of these plant species. And what I want to focus on for this talk is basically how common this red family is, the Sphingomonaceae. And another comparison family I've got here is the Pseudomonaceae, which includes Pseudomonas. And these are, of course, famous bacteria, not only beneficial, but also bacterial pathogens. And you see that their distribution is much more spotty, but the Sphingomonaceae seem to be quite consistent across all of these plants. And I should mention that in this we are only looking at leaves. So these are plant leaves, but they were consistently 15 to sometimes over 25% of the bacterial community. In another data set, not that one, we were looking at only Arabidopsis thaliana, and this is similar type of data. And again, bacterial families, and the colors are the same. And you see also that the Sphingomonaceae is quite common across many Arabidopsis plants from a different field site. And in this particular data I'm showing now, we classified the bacteria through metagenomics, meaning we didn't just amplify an amplicon of 16S and sequence that, we sequenced everything, including the plant leaf. And what that allowed us to do is to adjust those samples to look at the load of the microbes. And that process is shown by this animation. Basically, we get all the sequences, and of course, for every sample, we get lots of plant sequences from the leaf itself. And if we mathematically just scale those samples so that the plant is the same across them, then we can see for a given amount of plant, how do the microbial abundances vary. And when we did that with the same data set, what you can see is that the Sphingomonaceae, they're quite common and quite consistent across all the plants, whereas the Pseudomonaceae, they are very abundant on some plants, but almost absent on others. And the way to summarize that is through this chart, which shows for bacterial genera on Arabidopsis, on the x-axis, the coefficient of variation, which is a measure of how consistent the bacteria are represented. And you can see that Sphingomonas is on the far left, meaning it's very consistent across plants. And then on the y-axis, we have the bacterial load. How abundant is it overall? And you see that Sphingomonas is overall one of the most abundant genera, along with Pseudomonas. But the fact that it's here, it's abundant and it's quite consistent across plants, that particular feature is especially interesting for something to study that's successful in the field. It's able to colonize many plants to quite a high level without causing disease. And that's an interesting feature for a microbial inoculant. Another thing that we were able to take advantage of for Sphingomonas and also for Pseudomonas is the fact that these can be easily cultured on aerobic agar media. And in fact, with simple antibiotic can culture quite well selectively each of these genera. So we did that from these plants from the field site for both genera. And what we did to see if the culturing was really true, if it was really biased or not, was we cultured directly. I mean, we sequenced directly from the plant material and classified what 16S sequences we got. So these bars represent different 16S, different strains with different 16S sequences. And this is based on uncultured direct sequencing. And then we also cultured 410 of these isolates. And for each of the cultured ones, also looked at the 16S sequence. And what you can see is these distributions, they match quite well, which means that the culturing is not biasing the diversity of the Sphingomonas that we were able to recover. And it was a similar story for Pseudomonas. Pseudomonas was much more dominated by one particular strain group, but for culturing, we're also able to get many of the other 16S sequences there. So the culturing is something that we, for this work and for future work, we exploit a lot. And that, of course, allows us, if we're able to culture them, then we can also make pure DNA from each of those strains quite easily and sequence it in a very similar sequencing method to what GetGenome offers. And in this case, we sequenced many of those Pseudomonas and many of those Sphingomonas. And one of the first things I thought was quite interesting about the Sphingomonas is if you compare the genomes of Sphingomonas to each other, they're really quite diverse compared to if you do the same thing just with the Pseudomonas. And that's what I'm showing here. Basically comparing very like Pseudomonas genomes that all have the same 16S sequence to all the other Pseudomonas genomes that have that same 16S sequence. And that's shown, the Pseudomonas versus Pseudomonas comparison is shown in this dark blue box. And MASH is a quite nice bioinformatics program that compares genomes just by comparing short sequences called k-mers. And if two genomes are the same, all the k-mers, all the short sequences match up pretty well, and you get this high score. And this program runs really fast. That's why it's really nice. And you can see that the Pseudomonas are very similar to each other. But when we compare the Sphingomonas genomes to the other Sphingomonas genomes, even though they have the same 16S sequence, we see that these scores are quite lower. And this corresponds quite well to the average nucleotide identity. So you see some in this 80 range here. So despite having the same 16S sequences, some of these genomes have 20% or so nucleotide differences. So there's a lot of diversity there to be studied. We also looked in these genomes at different features that are known or hypothesized to be important for biotic interactions. And some of these features would be secretion systems that allow the bacteria to put proteins or other molecules into other bacteria or into host cells, either for conjugation or bacterial communication or communication with the host. And we could see that basically if it's dark, it means it's there. This is the tree of our 410 Sphingomonas isolates. And what you can see is that there's quite a few that have the type 4 secretion system represented, quite a few that have the type 6 secretion system represented, type 2 as well, not anything really clear with the type 3 secretion system. There's also, interestingly, there's some of these bacteria can photosynthesize on their own, some of their own carbon, so they have the full pathways for anoxygenic photosynthesis, and basically they all have flagella. There's so many, this is a difficult kind of figure to make because and I think many of you that work with genomes struggle with this too. It's like, there's so many different things you can look at, like what, but I decided here just to focus on some basic biotic interaction systems. And I mentioned that culturing is very powerful. Another way we've exploited culturing is to make what I call bulk culture metagenomes. And that's a way you can study a whole population of bacteria quite easily. And the reason that this is important is if you want to study a population of Sphingomonas on a plant leaf and you just make DNA from the plant leaf and you try to sequence that, you get so much plant leaf back that you waste your money and you can't really see the Sphingomonas genes that you want to see. But if you first played out the Sphingomonas on an agar plate that represents the community that was on that leaf, then you can scrape it off into a pool, make DNA from the pool and sequence that. And then you get a whole pile of genes that represents all the genes in that population. And the approach I took to analyze that was to make these metagenome pools and then map that data back to all the individual genomes. And what that can tell you then is if the data if the data is mapping to at a high level to one of the reference genomes, then likely that reference genome is represented in that metagenome. So here you see on the top, these are Sphingomonas reference genomes. And if we look across, each of these represents a sample from each tiny row here represents a sample from one of these plant taxa in the spring or in the summer. And you see like certain areas where it's dark. That means that, for example, this group of strains was found in these plants. Basically, the same group of strains was found on several plants in the spring, but not so much in the summer. So these patterns start to emerge. And one of the conclusions that I took from this was that Sphingomonas is also quite general in what it can colonize. If we saw, for example, that this was black, and then this was black, and then this was black, so basically that each plant was hosting different Sphingomonas, then it would be not general. But the general is kind of something also that's nice. And if you're looking at agricultural inoculant, basically the strains are able to colonize quite diverse plants. Another reason I got interested in Sphingomonas is because some of the strains can protect plants from pathogens. And I'll just show you one experimental example from that we did. We grew Arabidopsis in agar plates, and we exposed the seedlings to Pseudomonas pathogens. And when you do that, so here's seedlings grown for 2 weeks without any pathogen, so they grow pretty nicely. If you give them a classic Pseudomonas pathogen, DC3000, they're clearly smaller. And if you give them this other pathogen we got from the field, they basically disappear. They're just liquefy. And some of the strains, not many, it's a minority, but some of the Sphingomonas strains we tested are able to prevent this from happening. We don't know the mechanism of this, why yet. This is one of the kinds of things we would like to learn. But what I can tell you is, like from a published example that I thought was especially cool, how it can work. So this is a group that was looking at a Sphingomonas bacteria colonizing rice seeds. And they found that this can protect from the pathogen that causes seedling blight. And what the Sphingomonas can do is that not only does it degrade the toxin that the pathogen makes, and basically it eats the toxin the pathogen makes, but it also makes, the Sphingomonas makes its own small molecule that can turn off toxin production in the pathogen. So those are two cool molecular mechanisms, but I think there's all sorts of ways that it can happen. As I said, it's a very diverse genus. So what I'd like to just tell you a bit now is just give you a little bit of overview of what we're trying to do in Sweden. And we're continuing, of course, with culturing and using the sequenced culture collection. And some of the reasons for that will become clear also in future slides. And we're working with plants that are important to Sweden. Of course, Arabidopsis, because it's easy. But we're also starting to work with barley, with a model tree aspen, and Norway spruce, which is actually one of Sweden's most important crops. The whole northern part of the country is just covered in forest that's managed and processed for lumber and paper pulp. And so it's extremely important for research here. One thing we noticed when we started to close genomes with longer reads is that the plasmids are quite a common feature. What I'm showing here is for several of our genomes that we closed. The orange dot represents the main bacterial chromosome, so they tend to be like around 4 million bases. But many of the genomes have smaller circular elements, or plasmids, that up to 5. And we don't know exactly what they do yet, but we see that they have various secretion system components or lipid transport, antibiotic resistance. And this is actually one of the my PhD student, Sunta. This is one of the goals of his PhD, is to understand what the bacteria plasmids contribute to their fitness. And this involves some genomics, but also functional tests. Like, we want to cure these strains of the plasmids, means get rid of the plasmids, and then see how do these strains survive in various plant habitats. for example. And what I want to just lay out as kind of a brain teaser, something we want to try, actually. One thing that's important to us is we want to generate a large culture collection. We want to get the sequences of the plasmids. And if we're using short sequence reads of 150 bases, we can't be so confident, actually, that we have the plasmid because the assembly is not clear enough. So we want to assemble these genomes with long reads, but if we want to sequence lots of genomes, so like our goal is 1,000, with typical long read library production strategies, this would be just enormously expensive. And if we want to barcode many strains and put them on a machine like Pacific Bio, yeah, it's too much money. So the strategy that we want to try is essentially we'll culture all of our strains. We're going to use a low-cost method to make short read genomes that have all the genomic information, but they don't have enough information to know which are the plasmids. And then we're also going to take a pool of all of these strains and just sequence them as a pool, unbarcoded on a PacBio instrument. And then use the combination of the short reads that are barcoded, that we do know which strain is which, mapped to the long reads, or vice versa, map the long reads to the short reads. And then because these long reads will allow us to assemble the plasmids, then we can basically take advantage of the long read sequencing without paying for all this library prep. So I think it's possible then that we'll be able to do this quite cheaply, but I'm sure some of these details are more comprehensible to those of you that are really working with these systems, but I just thought I would throw that out there. We're going to try it. I hope it will work. And also, we're using various genetic approaches in Sphingomonas, and I think this part is maybe particularly interesting for this GetGenome group, because I know that as a GetGenome, you will get a typical project between 1 and 10 genomes back. And so what can you do with a model bacterial genome besides analyze the genome? Well, one thing that you can, of course, do is then use that for various mapping strategies. We are setting up a genetic screen in one of our model strains. And I think this works with this is not something we invented, but I think it works in an extremely cool way. So I just want to explain how, first, how that works. And we're using that for random mutagenesis. And we're also working with some reverse genetics to test and knock out genes that we hypothesize are going to be important. And I think this is also potentially of interest to this crowd. Co-transcriptomics is something that I won't talk about today. It's something we also are working on, though, to understand how the bacteria, what genes do they turn on the plant, and likewise, what genes does the plant turn on. So this genetic screen is something developed by the Deutschbauer group in UC Berkeley. And it's a variation of a very classic method of mutating bacteria, which is TN-seq. And that works by you take your bacteria, you mix it with a population of E. coli that carries a plasmid that has a transposon. And what happens is then that transposon, when it goes into the strain, the Sphingomonas strain, it jumps in each one to essentially a random location. And in the process, it disrupts the bacterial gene. But the cool thing about these transposons is not only do they disrupt the gene, but every single transposon has got its own genetic barcode, which is a 20-base random sequence. So the cool thing is then, sorry, I should just say, so you do this on a very large scale. You mutate, you collect many, many thousands of colonies where each one represents a different mutation. And so Ezgi, my student, did this on huge plates, and so 32 of these plates, and then you collect all of those, and that's your mutant pool. And the first step is you sequence this region. You get the barcode of the transposon, and you get some of the bacterial genome. And now if you have the reference genome, you can use this section to map each barcode to where it went in the reference genome. But then you only have to do that once for this whole population. Now to do an experiment, since you know where in the genome every barcode is, you can just use PCR of the barcode alone, and that will tell you which mutants you have in your pool. So then the way you do an experiment is like this. You put the population on something like an agar plate, you get your mutants back. You put them on the agar media, emit the mutants back. Let's say you put them on the plant, some of the mutants are missing. Those are then the interesting ones because those have potential mutations in genes that were important to grow on plants. So you do these kinds of experiments enough and you get a list of hypothesis genes that are important to grow on plants or grow on plants in different conditions. And we can follow those up with reverse genetics. So this is something that we're in the middle of at the moment. I don't have candidates to talk about with you. But once we get those candidates, we want to create specific mutants. And there's various ways to do that. And I think this next method is especially cool. It's something that came out last few years, and it's CRISPR-guided integrases for bacteria. And what it does is essentially lets you target a transposon to any part of the bacterial genome you want. And it works in quite a few bacteria. And you might find that it works in yours. It worked in Sphingomonas, and the authors of this method have found that it works in quite a few others. And so it's a single plasmid. You can put something in the payload. So we're using tetracycline resistance, and we're also putting a barcode there. And then you put in your guide sequence. So we've started by knocking out carotenoids, and those are what the pigments that make the Sphingomonas yellow or orange. And we're also interested, what is their role in fitness? And it's also very easy to see if it worked. For example, if you use that on one of our model strains, you can clearly see after we knock out the carotenoid pathway, the bacteria are white. And we've checked that this basically works as advertised. You usually get one insertion in the right spot. So this is a powerful strategy, not only because it's quite easy, but it allows you to extend the capability of whatever molecular biology and cloning you've done. Like if you want to make a mutant using another method like homologous recombination, probably you have to make a separate construct for every single bacteria that's a little bit different from the other one. But if you're only asking that several bacteria have the same 32 base sequence, that's easier to find. So if I take this diverse group of Sphingomonas, just using 10 guide sequences for a region downstream of the GLMS gene for tagging, you can get all of these. So that's just 10 guides for like over 100 strains. It should work in principle. And same for these other genes. It's quite easy to find these short sequences. So you can make constructs that are good for mutating more than one thing. So that's kind of a cool thing that you can do then when you have this system and you have the genome sequence of your organism. Last thing I want to just touch on briefly that we hope to get more into soon is that Sphingomonas is a particularly unique bacteria in that its outer membrane is made differently. It has, instead of lipopolysaccharide, which is the dominant molecule on the outside of gram-negative bacteria. Gram-negative bacteria, just to remind you, they have a double layer membrane. And it's this outer membrane that's covered with lipopolysaccharide in most bacteria. There's some bacteria, though, that can make what are called sphingolipids. And although these are rare in bacteria, they're extremely common in mammals and plants and all other eukaryotes and fungus, and they're essential. So what do the bacteria gain from that, actually? It's known that fungus that interacts with plants, the fungal sphingolipids can be recognized by the plants, or at least oomycete sphingolipids can be recognized by the plant as virulence factors. But for bacteria, plant-associated bacteria, it's still not known what could the sphingolipids do. So I think that the fact that these bacteria are so common, definitely it would be just so strange if the sphingolipids are not perceived somehow by the plant. So I think this is a potentially interesting signaling molecule, or a way that somehow the bacteria are able to not trigger such a strong immune reaction or something like that. So, I mean, the kind of experiments we want to do are, for example, to purify some of these sphingolipids and apply them to plants and just look at what is the plant transcriptomic response, for one thing. But in a project that I hope to get funded, we also want to manipulate the genomics of these sphingolipid pathways and try to make custom lipid configurations, and also see what is the role of this on the bacterial fitness. This is just some work by Hugo. We just did a very simple mass spec analysis of the sphingolipids on several of our strains, and it's quite clear that the composition of various strains in our collection is quite different and dominated by different sphingolipids. The last slide I have is a project outline just for a postdoc who started very recently, but I wanted to show it because it represents some of the more practical side of what we hope to do. There's a really bad problem with a fungus called Heterobasidion that attacks Norway spruce. That's the main agricultural tree here. It's the quintessential Christmas tree. But it's used here for paper pulp and things like that. And this fungus causes heart rot of this tree. And there's all sorts of strategies to try to prevent it, but there's a lot of interest in biocontrol, using microbes to stop that. And one product on the market, there is a Pseudomonas from soil in Germany somewhere that seems to work okay. But what we want to know is basically, does the bacteria from a soil in Germany, is there an advantage actually if we isolate Pseudomonas or Sphingomonas directly from Swedish Norway spruce? It's quite easy to hypothesize that the bacteria living in the plant might be adapted somehow to this spruce environment. So we want to basically then test home and away strains. Do the home strains colonize the spruce better? And if we give them the pathways necessary to combat this pathogen, for example, if we clone in a Pseudomonas pathway known to combat this pathogen, can the local strains work better? So anyway, that's just an overview of what we're trying to do. The group's now only been around, well, now around 2.5 years. So I feel like we're kind of turning the corner where projects are coming together, but there's not a lot of I say new results from the group to show you, but there's a lot of excitement. So, and hopefully, maybe some future collaborations with some of you. So with that, I will close. I'd like to thank all of you for listening, and James and GetGenome for letting me talk. I'd like to thank some of the people who helped a lot with the work from Germany, Prachaya, Rashe, and Julian especially. And this is my current group then. And yeah, it's been quite a transition to go from postdoc to leading a group, but they've made it very, very nice. And then various funding sources that have funded this work through the years below. So thanks. Take any questions. I should mention that this picture was taken from outside the apartment. So Sweden's very cold, but there are some beautiful things here. So if you ever get to visit, it's not that uncommon. So anyway, yeah, take some questions.